Okay, so jumping into population ecology, there are going to be a few different things that we talk about. To start with, though, I think the first thing to talk about is to talk about how we can assess or think about the health of populations or just tracking them in general. Okay, so we're going to start with talking about population density. So population density simply is defined as the number of individuals per unit of area or sometimes volume. Okay, so population, that's an abbreviation for the word population. Um, Population density is the number of individuals per unit of area or sometimes volume, right? So if you were a fishery biologist, fisheries are what we refer to as populations of fish that people like to catch and eat. Okay, so if you were a fisheries manager, you would be concerned with the population density of mackerel. I'm picking a fish, right? And so you would want to know how many mackerel live in, oh, I have a visitor. It's like there's something behind me that's moving. Come here, Luigi. This is Luigi, my polydactyl kitty. Look at his foot. Look at that. Look at all those toes. He's got so many. I know you didn't know I was going to bug you. That's what you get for visiting me. Mario will not come in here while I'm doing this. Mario is a love bug, but he does not like being on camera. Anyway, that was Luigi. Okay, so if you're concerned with managing mackerel populations to make sure that there's enough mackerel for people to enjoy for, generations and generations, right? You want to have good tabs on how many mackerel there are, okay? And so the way that we quantify that is number of individuals per unit of area. Like I said, sometimes volume. If you're talking about like earthworms or something, instead of looking at like a square meter, you might be looking at a cubed meter. So you're looking at, two directions this way and then depth too. Usually it's area though, okay? How do we quantify population density? Usually, the vast majority of the time, we do not count population density, I can talk, by literally counting every individual. Okay? There's a variety of reasons why. One is it's hugely impractical and it's a waste of time, right? So could I say, I need to know how many Jeffrey pines there are in, within the city limits of Wrightwood? I'm just making up something stupid, right? Sure, somebody could count that, but that is totally impractical and lame, okay? Another reason that we often don't do direct counts like that is that, particularly when we're thinking about animals, animals don't generally like sit there and be like, I'll wait for you to count me and I won't move, right? I'll just, I like people. I'll hang out, and let you count me before I go about my business. Yeah, that doesn't really work. Okay. So there are lots of ways that we estimate population density. And so that can be things like estimating, if let's say you wanted to know my question about how many Jeffrey pines are there in Wrightwood. Okay. What I might do is I might choose several acres of land in Wrightwood, and I might count the number of Jeffrey pines in each of those acres. and then like extrapolate, just sort of say, okay, within these 100 individual acres that I chose, there were X amount of Jeffrey pines. So based on the entire acreage of Wrightwood, I can, assume there's an estimate of approximately blah, blah, blah. Okay. So we often do estimates like that with organisms that hold still. Okay. So plants and other certain animals that don't move. So one of the things that I find enchanting is this idea of what it means to be an animal. And a lot of people have this really sort of biased idea of what an animal is by thinking about what kind of animal you are. Okay. And so one of the things we are going to explore in our organism of the week is we're going to explore, that what you think isn't an animal, what you think an animal is, a lot of animals are not, right? And so we're going to explore that a little bit, right? There are some animals that don't move, like, at all. There's lots of animals that don't move much at all. Okay. And a lot of you think that, moving their body from one place to another place is a, main feature of being an animal, right? So anyway, we'll talk more about that in another venue, not in this video. Okay. So how do we count things that are problematic? One way is by using indirect indicators, and that's what this picture is about. So these little guys are prairie dogs. Prairie dogs are rodents that are heckin' adorable. They are very common in the Great Plains of the Western US. And so for a while in my childhood, I lived in Colorado, and we lived, kind of next to the foothills of the Rocky Mountains. And so on one side, giant mountains, on the other side, flat plains, right? And we had prairie dogs all over the place because where I lived at the time anyway was not very developed. There were, like neighborhoods and communities that were popping up, but there was also just a lot of like open prairie. Okay. So how do you count prairie dogs? Well, one way of doing it is if you have some idea of, how many prairie dogs typically live in a burrow, right? So we know that prairie dog families are typically, 6 individuals. I'm making that up. Okay. One really quick way of estimating the population size through this method of indirect indicators is by counting their burrows, right? And you can do that even like in a plane or a helicopter, right? So fly over a prairie and just with a little clicker, those little clickers that bus drivers have to count people on the bus, right? Use a little clicker to count the number of burrows that you see. And if you know the standard prairie dog burrow has 6 prairie dogs, boom, now you know how many prairie dogs there are in that area. Okay. So indirect indicators. We use this with burrows and nests, bird nests, right? So when I was in college, I did some research on birds in Camp Pendleton, right? And one of the ways that we estimated population density of all kinds of different species of birds was by counting their nests, right? So how many nests do you see? We know that each nest has to have, at least in the case of birds, has to have a mommy and a daddy bird, right? And then if, for any given species, we know how big their clutch, that's what we call a bunch of baby birds. We know how big a clutch typically is, so we can get a feel for population density. Yeah, so that's indirect indicators. Another way that we can manage animals that tend to move a lot, right, and also don't really want us to count them, is a technique called mark and recapture. And so this is a good one. These little birds are being captured in a device called a mist net. And so you almost can't see it in the picture because that's what they look like. They're hard to see. And this is kind of mean, but when we do these types of things, the reason that we do them is ultimately to, benefit the animals, to understand about, how their population is doing and all of that. So, it's always to benefit the animal, but, sometimes they have temporary irritation or disruption, right? So the way that you use a mist net for birds is many species of birds spend their day foraging for food, right? And then they roost, so they sleep overnight in a tree or some location, right? And so if you set up a mist net that is in between where they're foraging and where their roost is, and the net is very light and sheer, the birds can't see it. So they fly right into it and then they get tangled, right? And then you carefully untangle them out of the nest and collect some information about them. So, in addition to just how many do you catch, right? Because population density, that's what we're focused on. You also might collect other data like, what is the sex of this bird? Is it a male or a female? What is the size of this bird? Is it healthy? Is it a healthy weight? Is it light? Is it scrawny? Sometimes when we're doing studies like this, we also take advantage of the fact that we've caught an animal, and we do things like take a blood sample to learn about the hormones of the animal in that population, or take a DNA sample, right? A place on campus where we're actually really involved in doing some population ecology is Chafee's Chino campus has a colony of burrowing owls. I have a sticker on my water bottle. That's a burrowing owl. You need to Google them because they are the most adorable owls that exist. Okay, they're small and they're so cute. And so because they are what we call a species of special concern, which means they haven't been listed as being endangered, but they essentially are. We are concerned with making sure that the population is stable, hoping that the population grows. We definitely don't want the population to shrink, right? We're concerned about the health of the birds. We're concerned about all kinds of things related to these birds. And so at various different times, we do some different activities with a local biologist who specializes in capturing birds of prey. Okay, so he has all these different ways of like nets and stuff for catching the owls in a way that doesn't hurt them. And when he catches one, first you try to figure out what sex it is. Is it male? Is it female? It's kind of hard to tell with burrowing owls because it's not obvious. He gets the weight, he measures the length of the bird, and he pulls just, a couple of feathers, 2 or 3 little feathers right from their underbelly, right? Pulls them out, sticks them in an envelope, and sends them off for DNA analysis. So, you don't even have to, draw blood nowadays for DNA samples, right? You just pull a couple feathers and there's enough blood just in the little base of the feather, right? Enough living tissue there to do DNA, right? And find out all kinds of things about that population of owls. But the most important thing that you do when you're doing this technique is you have to somehow mark the individuals that you catch. Okay, so you see these little bands, see these little things right here on the little ankles of those birds. That's usually the way that we mark birds, is we put a little bracelet essentially around their foot, and they're made of plastic or aluminum. Typically they're very, very lightweight. Sometimes they're just colorful and we identify them based on the color. Sometimes there's information written on them like numbers. So the owls that we have captured at the Chino campus all have an identifying number. And it's large enough that you can actually see them. So if you're, out in the fields at Chino with binoculars and you see one of the burrowing owls with a bracelet on, you can actually, if you get your binos just right, you can actually see which one it is. Oh, that's M17. How you doing, M17? Good to see you, buddy. Right? I'm kind of getting off on a tangent, but the punchline is this: you mark them somehow and then you let them go. Okay? All right. So how does this tell you anything about the population size? That alone does not, right? So let's say you catch 10 birds in a mist net. So all that tells you is that you caught 10 birds. It doesn't tell you how big, because you didn't catch everybody, right? So it doesn't tell you how big the population is. Okay. Let's say you come back though a month later, right? You set up your mist net again. It's the same population of birds. It's the same group of birds that live in this particular roost over here. right? So it's the same population of birds, but this time I catch 20 birds. Of the 20 that I catch, 10 of them were marked from the previous capture, and 10 of them are new. Okay. Well, now you can assume if 10 of them are repeats and 10 of them are new, right? Then you can, especially if you do this multiple times, you can do a little pretty simple math actually to estimate the population size based on in subsequent captures, right? How often are you getting the same individuals and how often are you getting new individuals, right? So that's called mark and recapture. We do this different ways with different species of animals, okay? So with birds, it's little bands around their leg. With insects, you can actually write on them. So this butterfly has not been harmed, right? They're holding it in such a way to, protect its wings. And they've actually used ink to write B1 on the wings, okay? There's a deer with a collar, okay? We can also use all kinds of collars that like have radio tracking or GPS track. I mean, we can GPS track stuff now, right? So we know there are lots of species where, particularly large animals that have like large range where, you put a GPS collar on them or chip of some kind, and you can track them for their whole life, right? Using, satellite data. So, in the news, you'll hear about, oh, one of the mountain lions, and they give them numbers, right? This mountain lion was, killed, right? We found him dead because he stopped moving, right? We found him, or, he's moved to a new area, or, whatever. Anyway, okay, so we can do that too. So you can estimate a population size by capturing some, marking them somehow, letting them go, and seeing how many come back that you originally captured and how many are new. And that gives you a way of extrapolating, right, and estimating population. Okay. Another interesting, so that's just like, how do we do population density? Okay. Another interesting thing that we might look at with populations is we often care about how a population is dispersed. So we call these dispersal or dispersion patterns, sometimes distribution patterns, but essentially what we're talking about is we're talking about not just how many individuals they are, but how those individuals are sort of arranged in space. Okay, so this will make more sense as we go through the example. So there's basically 3 different dispersion patterns that you can have. One of them is clumped. The idea of clumped dispersion patterns means that for any given area, so this square represents an area, okay? You have X amount of species, X amount of individuals, X number of individuals within that population. But rather than being evenly distributed throughout, they're clumped together in bunches, okay? So why would that happen? Why would that be a thing? Why does clumping ever happen? So some of you are looking at this photograph of these fish, and perhaps you are a fan of Finding Nemo, and you'd like to go to the aquarium, right? And you know why some fish, not all fish, but some fish form schools. So why do fish form schools? Well, safety, right? So they all hang out together because each one of them is a little bit safer when they're in a big crowd than they would be if they were on their own. That's why Nemo shouldn't have left the reef. Come on, dude. What are you doing? What are you thinking about? Anyway, by the way, don't get any of your biology knowledge from movies in general, but particularly animated films. I was teaching a majors class one time, and a bunch of students were super confused. We were talking about jellies, jellyfish, and a bunch of students were super confused about something about like how they sting their prey and all of that. And I was like, I don't understand why you guys are so confused. And they were like, but in Finding Nemo. And I was like, oh yeah, no, Disney makes stuff up, man. That's not real. It was funny. Anyway, okay, so clumped could be for protection, right? Safety in numbers. What are some other reasons why you might be clumped? Well, let's say that you need a resource that isn't evenly distributed, right? So let's say that you are a large herbivore, like I don't know, moose. Okay. Well, they need to drink water, right? They like to eat plants that are, grow in really aquatic places. So you would probably find a lot more moose, around a lake than you would in a dry area. So they tend to clump around a resource, right? Or clump around a food resource or whatever. Okay. So clumped dispersion patterns are very common. Another dispersion pattern that's very common is uniform. It's essentially the opposite, right? So in this last one, it was like, okay, there's X number of individuals in an area, but they all tend to be in like these little piles and these little clumps. Okay. Uniform distribution is there's an X number of individuals, but they're spread out pretty evenly. Okay. So what are we looking at in the example here? We're looking at some beautiful penguins. Okay. Penguins are really cool because they, when they are nesting, when they're sitting on nests, they form an incredibly uniform distribution. So notice that the distance between this penguin and that penguin and this penguin and this penguin and this penguin and this penguin and that penguin and that penguin, it's like the same distance almost exactly, right? So in this particular example, it's about territory. It's like, this is my little nesting area. You stay in yours, you stay in yours. Okay, so this is territorial behavior. But you often also see uniform distribution when you have organisms that are competing for a resource, but the resource is not clumped. The resource is widespread. Okay, so another really good example of uniform distribution. You could see this, those of you that like to, drive places and drive through the desert, right? If you're going to Vegas or the river or whatever, right? When you drive through the desert, one interesting kind of thing that most people don't notice is if you look at the creosote bushes, for example, they often look like they've been planted in like a grid formation, right? Because their distribution is super uniform. And it's like, well, why would that be? Why are they so uniformly distributed? Well, they're competing for water. And so every plant has its little zone where its roots are, right? And it, is competing with other plants for water. And because the water's coming from the sky, right? There isn't necessarily, at least in the desert and, the types of deserts where you see this, there isn't like more water in this area and less water in this water area. So there's this really uniform distribution. Okay. So like I said, next time you go drive to Vegas, right? Look out the window a little bit, see what's happening. Okay, the third dispersion pattern is called random. I am sort of the opinion that this is BS. Random implies that it is neither uniform, nor is it clumped, but it's literally random. Some organisms end up being close together, some end up being spread out. It just, there's no particular order to anything. Okay, that's what random means. This sort of exists, but I'm sort of the opinion that it really doesn't, because there's always going to be a certain amount of clumping. There's always going to be a certain amount of random distribution. And so like one of the examples that of uniform distribution, one of the examples that they give is in textbooks typically is they talk about like clams in a mud flat when they're not mating as the tide is going down. It's like, okay, so that sort of exists for a while, but there are going to be other times when the clams are competing for food, or the clams are clumping together because they're looking for a mate or whatever. So, random happens, but it's not common. Okay. All right. So some examples. What dispersion pattern are you seeing here? What is this? What are these? What are these? This is a different kind of penguin, right? And look how many there are, as far as the eye can see. And they're all uniformly distributed. Clumped. Okay. Like I said, uniform and clumped are kind of the two big ones. One other really interesting thing to think about is dispersion pattern is very much affected by scale. Okay, so here we're looking at a couple of different interesting pictures of humans. What is the dispersion pattern like of humans, right? We're animals. How we can look at our dispersion patterns too. So I love this beach picture because we have all been there, right? You go to the beach and you don't want your, towel and your stuff too close to anybody else, right? So we're really sort of like territorial at the beach. It's like, and if somebody like plopped down next to you, if there was space over there, it'd be like, what are you doing? I don't know you, right? Like, very much we distribute ourselves in a uniform way at the beach. And that's how we build our houses, right? So that's what this picture is. It's showing a neighborhood where everybody has, the same size yard, same size house, same size yard. It's, very suburban, scary neighborhood. So on some scales, right, like the scale of looking at individuals or families or small groupings like that, we tend to distribute ourselves in a very uniform fashion, okay? But at a different scale, we tend to organize ourselves in a very clumped fashion, okay? So what you're looking at in that middle picture is you're looking at a satellite image that was produced where the purple color indicates amount of light, right? So visible at nighttime, okay? So light is a really good indicator of human presence because we tend to, have street lights and lights on everything, okay? And if you go to places where there are not a lot of people, there's a lot less light at night, okay? And so you can see very clearly, if light equals people, you can see that people are clumped around cities, right? So there's way more people in and sort of around Boston than over here, right? There's a ton of people in and around New York City, Long Island, right? Than there is over here, right? Providence, you might not have heard of Rhode Island, small city in Rhode Island, right? And so, yeah, so humans have both clumped and uniform distributions depending on what scale you look at. Okay, so it's kind of interesting. And other animals do the same thing, and plants for that matter, right? Plants can be clumped, plants can be uniform. Okay, so I'm going to stop here, and then the next short video is going to be about population growth. Okay, so more population ecology to come, y'all.